I'm interested in the role of central thalamus, which is a part of the brain that's been proposed as a site for stimulating cognitive function, particularly in diseases like schizophrenia or cognitive disorders that produce severe impairment. Central thalamus has been proposed as a site for stimulating cognitive function based on its role in regulating arousal and awareness in the brain and its connections with parts of the brain that have very important roles in different cognitive functions. We're interested in working memory, which is the ability to hold information in short-term memory while performing tasks like making a decision. We've conducted a number of lesion studies that have identified a particular part of central thalamus, the rostral interlaminar nuclei, as a critical site for working memory. The work I'll be talking about focuses on treatments that are designed to either increase or decrease activity. In some experiments, we micro-inject drugs into that part of the brain. Some of the drugs decrease activity, and we found that these tend to impair memory in a dose-dependent fashion with greater impairment for greater doses. Other drugs, the more interesting ones, enhance activity. And these we found evidence for an inverted U type function, which is consistent with the idea of hormesis, in that low doses tend to enhance memory function and higher doses tend to impair function. We've more recently started doing research in which we implant electrodes permanently in this part of the brain, and we stimulate the nerve cells with very small amounts of electrical current that sort of mirror or produce a response like a period of neural activation. This is a technique that's been used widely clinically to treat diseases like Parkinson's disease, dystonia. Recently, it's been used to treat patients with affective disorders who are resistant to other treatments, obsessive compulsive disorder, and in some cases, it's been used to actually treat patients who are in persistent vegetative states and have had consciousness reestablished, at least some behavioral recovery produced by this stimulation. What we have found in our rats is that low-dose stimulation can enhance memory performance, whereas high-dose stimulation tends to impair it. So it's very much like what we see with drugs to either inhibit or excite activity. What's interesting is that the timing of stimulation is very critical. So low doses at the time in which memory is being held or at the time that a response is being made is very critical. Whereas timing earlier or stimulation earlier in the trial tends to have very little effect on performance. So taken together, these results are consistent with a very old idea in psychology called the Yerkes-Dodson law, which suggests that arousal has a very powerful effect on complex cognitive disorders or complex cognitive abilities, that low levels or moderate levels of arousal are optimal. Whereas high levels of arousal tend to impair people, very much like the state that you get when things go bad on a tough day and you feel very frazzled and unable to sort of think or to function in a very coherent fashion. It's also very consistent with evidence in the literature looking at how stimulants can enhance function in ADHD or other conditions that affect attention or cognitive function, in that moderate doses of stimulants tend to be useful for enhancing function, whereas high doses tend to impair function.